Okay, this is the final segment of Chapter 10. We're going to discuss the Calvin cycle and some alternate pathways that some plants have evolved in order to handle. Now, just a little reminder. Remember, we talked about the light reactions, and remember, the light reactions take place in the thylakoid membrane, really, within the thylakoids. And remember, during the light reactions, there is an electron transport chain, which is going to produce ATP in a very similar way that occurs in the mitochondrion. However, there's a hydrogen ion gradient that is built up in the thylakoid space from the pumping of the protons from the stroma to the thylakoid space. And then again, the only way they can get back through to the stroma to get at equilibrium is via ATP synthase. And again, in the stroma is where ATP is made. And then during linear electron flow, electrons are going to be passed from photosystem 1 to ferredoxin, then to the NADP plus reductase, which is going to reduce NADP plus to NADPH in the stroma. So again, ATP and NADPH are made in the stroma, and they're going to be used by the Calvin cycle in order to fix carbon from carbon dioxide into organic chemicals like sugars. Now, again, we're going to kind of go to the overview here. Remember the light reactions. Again, I just talked about this. Involves the splitting of water, the release of oxygen, and the absorption of light in the thylakoid membranes, producing ATP and NADPH. NADPH and ATP are going to be used by the Calvin cycle, so ATP becomes hydrolyzed again, and then NADPH becomes reoxidized, and then goes back to the light reactions. But during the Calvin cycle, it is a cycle, so it's a continuous process as long as there is ATP, NADPH, and CO2 entering the cell. So carbon dioxide, remember, is a gas. This carbon must be incorporated into organic molecules in order to be used for energy. So the carbon is said to be fixed into organic chemicals via the Calvin cycle. And the Calvin cycle releases basically precursors to the sugar molecules. So they're really smaller sugar molecules, which will be used to make the larger 6-carbon molecules. And you'll see what I'm talking about when we get to that section. All right, so now we're going to get into the real details of the Calvin cycle. They're not as, I guess, complex as you might have thought for the light reactions. However, it can get a bit confusing. So I'll try to go slow. All right, so the Calvin cycle uses the energy from the hydrolysis of ATP and the energy that's stored as electrons in NADPH to reduce the carbon dioxide to a sugar. The first step is carbon fixation. Now, the Calvin cycle is very similar to the citric acid cycle in that it is a cycle, so there's a starting material that is eventually regenerated. So in the citric acid cycle, it's oxaloacetate. In the Calvin cycle, it's ribulose bisphosphate. The Calvin cycle is an anabolic process. Okay, so it's because we're building sugars here, and it takes energy to do it. And remember, the Calvin cycle uses the energy from ATP hydrolysis and the energy stored in the NADPH as electrons. Now let's talk about the Calvin cycle a little bit more in detail. Now, carbon is going to enter the Calvin cycle as carbon dioxide. We know that, right? So carbon dioxide. How it leaves is a little bit different. Okay, so carbon dioxide has one carbon. How it leaves is a 3-carbon molecule. Okay, so the 3-carbon molecule is called G3P or glyceraldehyde 3-phosphate, as I mentioned before. It is one of the intermediates in the breakdown of glucose in cellular respiration. However, we're using it to build glucose now. Okay, so this is actually what's released from the Calvin cycle. Again, it's a 3-carbon molecule, so then another one will be combined with itself, or another one, right? So 2 of them will be combined to produce a 6-carbon molecule. Now, as we know, carbon dioxide only has 1 carbon, but G3P has 3 carbons. So this means this cycle must take place 3 times in order to fix 3 carbons from carbon dioxide into this to produce this 3-carbon molecule. There are three steps in the process. Okay. The first step is the carbon fixation. Carbon fixation is catalyzed by a very large, probably the most common protein complex on Earth called Rubisco. After that is a reduction step and then the final regeneration of the ribulose bisphosphate, which is the carbon dioxide acceptor. Again, remember it's a cycle. So the starting material, which is ribulose bisphosphate plus the carbon dioxide, is going to be regenerated. However, the carbon dioxide isn't regenerated. It's the ribulose bisphosphate that is, because that's what's part of the cycle. Now let's look at the cycle. Textbook, it shows the cycle in reference to adding 3 carbon dioxides into it. So we're going to do 3 carbon dioxides all at once. Right? So what we're going to do is go through this cycle, but while we go through the cycle, we're going to count the carbons as we go through. What happens is, let's say 3 carbons or 3 carbon dioxides have entered the system, and Rubisco, right? So let's say there are 3 Rubiscos, one for each. So basically, Rubisco is going to take this carbon dioxide, these 3 carbon dioxides, and add them to a molecule, right? So this is the starting molecule. It's going to be a 5 carbon molecule, but there's 3 of them, right? So there should be 3 5 carbon molecules in which the carbon from carbon dioxide is going to be added. So now we have 3 6 carbon molecules. That's 18 carbons right there. Okay, so we have 18 carbons. Well, then this short-lived intermediate, right? So this 6 carbon short-lived intermediate is going to then be converted to 3 carbon molecules. So now there's going to be 6 of them. So 3 carbon molecules, we still have 18 carbons. It's just different form. All right. So these 3 carbon molecules are called 3-phosphoglycerate. Okay, so this is the carbon fixation step. The next step is a reduction step. What's going to happen is we have 6 of these 3 carbon molecules, 3-phosphoglycerate. They're going to be converted to 1,3-bisphosphoglycerate. So basically, a phosphate is going to be added to each one of them, making them more reactive. Okay, so here we're going to use 6 of those ATPs that are made from the light reactions. By hydrolyzing them, we're going to use that energy by hydrolyzing them, creating another phosphorylated intermediate. Okay, so then this is going to be reduced. Okay, so this is the real reduction part. Okay, so the 1,3-bisphosphoglycerate is going to be reduced. And remember, oxidation reduction occurs together simultaneously, so they're coupled. So then if this is reduced, then something must be oxidized. And since we have 6 of these molecules, 6 NADPHs are going to be oxidized to NADP+, and then releasing 6 inorganic phosphates. This is going to result in 6 G3Ps. Remember, I told you it's G3P that leaves the cycle, right? So remember, we've incorporated 3 carbon dioxides to make, ultimately we're making 6 G3Ps, but only one of them is going to leave the cycle. Okay, so it takes 3 carbon dioxides, 6 ATP, 6 NADPHs to produce only 1 G3P. That's not even a full glucose molecule. That's a lot of energy. Okay, so here we have 6 G3Ps, only one of them leaves the cycle. The other 5 are going to be used to regenerate the carbon dioxide acceptor, okay, which is RUBP up here. So basically, we have the carbon dioxide being combined with RUBP to make that short-lived intermediate. And Rubisco combines the two. Regeneration of the RUBP involves the hydrolysis of 3 more ATP because, as you can see, RUBP has a phosphate on both ends. And here we have 15 carbons. Remember, we had 18 here, 3 of them left. These are going to be the sugars that are going to be ultimately making glucose. And then we have 5 of them left, so that's back to 15 carbons. And then there's a few steps, and basically the ATP is hydrolyzed, and then now we have 3 RUBPs back to 15 carbons. Okay, then if we continue the cycle again, we're going to combine the RUBP, the 3 of them, with 3 more carbon dioxides to make 3 more of that short-lived intermediate, resulting in 18 carbons again. Okay, so then again, we're going to lose 3 and then back to 15. And then 18 again, we're going to lose 3 and back to 15. Again, these G3Ps are going to leave the cycle, and then they're going to be used to build other carbohydrates, much larger carbohydrates, like 6-carbon carbohydrates, and then ultimately starch, which is stored in the chloroplast. All right, so just to kind of review, we're going to go back to this. What happens here? We have the light reactions. Remember photosystem 2, the electron transport, photosystem 1, and then the electron transport between photosystem 1 and photosystem 2 with photosystem 1. So this could involve the linear electron transport or just cyclic electron transport. However, it's linear electron transport that produces the ATP and the NADPH for the Kelvin cycle. So water goes in and it gets split, light is absorbed, and then a bunch of chemical reactions are happening here. What leaves is oxygen. And then ATP and NADPH are produced, which are going to be used by the Kelvin cycle to fix carbon from carbon dioxide, which is going to enter as the oxygen is leaving. And then what leaves the Kelvin cycle is glucose or sucrose, right? So we have sugar molecules that are going to be produced with the actual G3P that leaves the Kelvin cycle. Okay, so that's ultimately the process of photosynthesis. Now, there are some alternative mechanisms that some plants have evolved in order to be able to survive in hot, dry climates. Remember, photosynthesis requires water, right? So plants require water because really that's the first step. Water is split. What if it's hot and dry? Most plants wouldn't be able to survive those types of conditions. However, there are some plants that can. Now, it's really alternative mechanisms to the fixation of carbon that has evolved. Okay, so it doesn't have anything to do with really the light reactions and water, other than the fact that water can be limiting under those conditions. It's how they fix the carbon. Now, again, dehydration is going to be a problem for plants, especially those that do not have any alternative mechanisms to fix carbon under conditions where, water is limiting. So on hot, dry days, plants typically close their stomata to conserve water. But unfortunately, this limits photosynthesis, right? So if water's not coming in and the stomata are closed, right? So they're trying to conserve water. The stomata are closed. This is limiting the amount of gases that can go in, right? So this limits the entrance of carbon dioxide. And then this results, since the stomata are closed, the oxygen that's inside the plants can't leave. So now we have a situation where there's low amounts of carbon dioxide, however, high amounts of oxygen that are starting to build up inside the plant. These conditions are very wasteful, okay? The process of this is called photorespiration. Now, photorespiration, the question is whether it's an evolutionary relic, right? So it's why is that question posed, right? Because we know that there are plants that actually have evolved to be able to get around these types of climates, to be able to survive in these types of climates. Okay, so it's thought that, of course, photorespiration, this process, might have kind of triggered the adaptation of these plants. All right, so in C3 plants, or most normal plants, right? So that's what we've been talking about. So whatever we discussed right now and already about photosynthesis, we're talking about C3 plants. The initial fixation of carbon dioxide occurs via rubisco. Okay, so rubisco initially fixes the carbon dioxide and then forms 3-carbon 3-phosphoglycerate. So remember, that was that step where you had that short-lived intermediate and then the 3 carbon molecules. Then you had 6 of them, you had 18 carbons total. That's why it's called a C3 plant, because the carbon is fixed into 3 carbon compounds, and these compounds are called 3-phosphoglycerate. In photorespiration, because oxygen is starting to increase because the stomata have closed, then oxygen is going to be incorporated into, instead of carbon dioxide, right? So oxygen will bind Rubisco. Okay, so Rubisco is not very picky. Rubisco is a complex, a protein complex that can bind carbon dioxide and bind oxygen. Now, it's more likely going to bind oxygen when carbon dioxide is very low. Okay, this is going to produce two carbon compounds. Okay, these two carbon compounds are going to be very wasteful because it doesn't allow the plant to make the sugars, which will be required to for energy, right, later. Photorespiration is going to consume this excess oxygen that's building up inside the plant and the organic fuel, which is going to be ultimately releasing the carbon dioxide without producing ATP or sugar. So no ATP is going to be made, no sugar, the plant's losing energy, it's not producing sugars because it's not incorporating the carbon dioxide, it's incorporating the excess oxygen that's building up inside the plant. Those are all conditions which are very, very wasteful. Now this kind of shows that process. Rubisco is really at the center of it all. Okay, so remember the Calvin cycle. Carbon dioxide is incorporated. Carbon dioxide binds with Rubisco and RUBP to produce ultimately G3P, which will leave the Calvin cycle. Okay, now when there's excess oxygen and the stomata are closed, photorespiration is going to occur. So Rubisco is going to bind to oxygen and RUBP, producing 2-phosphoglycolate and 3-phosphoglycerate. Then ultimately ammonia and carbon dioxide is going to be released. ATP continues to be hydrolyzed and the plant will eventually die. Now this is C3 plants. Other types of plants have adapted other mechanisms to circumvent the incorporation of the oxygen into the Rubisco. Right. C4 plants are pretty special. C4 plants, they have an alternate mechanism to kind of bypass this photorespiration process by incorporating carbon dioxide into 4 carbon compounds, and that's why it's called a C4 plant. Now, it doesn't use Rubisco initially, okay? They have what's called a spatial adaptation. So there's 2 cells that are involved in the fixation of carbon and then the production of sugar. Okay, so spatial means geographical difference, right? So it's separated into 2 types of cells, right? So this shows the leaf anatomy of a C4 plant. We're going to look at it a little bit closer. However, you can see the vascular tissue, which adds the water from the ground. We have the stomata, which are going to close during a hot, dry climate. And then we have the mesophyll cells, which are also in the C3 plants, but then we also have the bundle sheath cells. Okay, so the C4 plant has also the bundle sheath cells, and you can see the mesophyll cells and the bundle sheath cells are adjacent to one another, so they're very close, and that's going to become important. So let's look at a little bit closer. So here we have the mesophyll cell and we have the bundle sheath cell. Now, what's different about the C4 plant is that the carbon is fixed initially in the mesophyll cells. However, it doesn't involve Rubisco, which is going to be present in the bundle sheath cells. So C4 plants have this, this enzyme called PEP carboxylase. This is a special enzyme. Now, when photorespiration is occurring, remember, oxygen is building up and very little carbon dioxide is entering because little to none, because remember the stomata are closed and they're trying to save water. Okay, so they're trying to conserve water. Now, PEP carboxylase isn't like Rubisco, where Rubisco can bind oxygen when it's in high quantities. It can also bind carbon dioxide. However, if oxygen is high enough quantities, it can bind it instead of carbon dioxide. Well, PEP carboxylase has a greater affinity for carbon dioxide. In fact, it really doesn't bind to oxygen. So it doesn't matter whether oxygen is in very high levels. PEP carboxylase will still bind to whatever little carbon dioxide is entering the plant. Okay, so here, PEP carboxylase is the enzyme that actually initially fixes the carbon into 4-carbon compound. And remember that from the citric acid cycle, the oxaloacetate? Well, instead of regenerating oxaloacetate, we're making oxaloacetate by adding carbon dioxide rather than releasing carbon dioxide. So we're kind of going the other way. So again, remember malate, right? So oxaloacetate is converted to malate, and then malate is going to be decarboxylated, whereas the malate is going to lose its carbon dioxide in the bundle sheath cells. When malate loses that carbon dioxide in the bundle sheath cells, it becomes pyruvate. Remember pyruvate? Okay, so then some ATP is hydrolyzed and pyruvate is converted to PEP, which is then basically regenerated so that it can combine with the next carbon dioxide via PEP carboxylase. Okay, so that's the initial in the mesophyll cells, the initial carbon fixation. Well, what happens to it after that, right? So remember, malate lost that carbon dioxide. So basically, took the carbon dioxide from the outside via PEP carboxylase and then basically transferred it to the bundle sheath cells where rubisco is. Okay, so this is concentrating that carbon dioxide into where rubisco is going to be so that rubisco binds to the carbon dioxide and then goes through the typical Calvin cycle to produce sugars, and then of course transporting the sugars to the vascular tissue so it can be transported throughout the plant. Okay, so that's the C4 plant. This is the C4 pathway and involves the initial fixation of carbon via PEP carboxylase. Well, there's another type of plant. It's called a CAM plant. So the process of a CAM plant is called crassulacean acid metabolism, or CAM. Okay, so this is a process which allows the plant to fix carbon. Okay, this is a temporal adaptation. So temporal means different times. Okay, so different time of day that things are happening. Now, CAM plants also have PEP carboxylase. Okay, but what CAM plants do is keep their stomata closed during the day to conserve water. Okay, so when they keep their stomata closed during the day, the only thing that's really happening there are the light reactions because, again, it's during the day and that's what the light reactions require. And then what happens here is basically oxygen is going to be produced during the light reactions. However, the stomata will not stay closed. They're going to open them at night. Okay, so when they open them at night, that's when the carbon dioxide can come in and the oxygen can leave. Now, yes, oxygen will build up during the day, but remember, they also have PEP carboxylase. So PEP carboxylase is going to bind to whatever little carbon dioxide that is present during the day, and then all the carbon dioxide at night, where the Calvin cycle is going to be functioning at full force. Okay, so really, they're going to conduct the light reactions during the day and the Calvin cycle at night. So these are two adaptations. So these are some examples. So an example of a C4 plant would be sugarcane. It can live in those hot, dry climates. Pineapple, also, this is a CAM plant. So again, the C4 plant has a spatial adaptation. The CAM plant has a temporal adaptation. The C4 plant has the two types of cells in PEP carboxylase. The CAM plant also has PEP carboxylase. However, what it does is basically different processes of photosynthesis are going to occur at different times of the day. Okay, so just kind of a review. We've talked about this already. Life either directly or indirectly depends on photosynthesis. So directly meaning that those organisms that need photosynthesis to produce their own food. And then, of course, indirectly is going to be where we have organisms like us. We actually depend on photosynthesis to produce food, which we will take in or ingest. So we've seen this before. Remember, the water kind of comes from the ground, and of course, when it rains, it will be transported up through the vascular tissue into the plant to the leaves, where it will become split. And this point, the oxygen will leave, and carbon dioxide enters through the stomata as the oxygen leaves. And then, of course, with the presence of light, the light reactions are going to occur. And then remember, the light reactions are going to produce ATP and NADPH, which will be used by the Calvin cycle to fix the carbon into organic molecules like sucrose and then be transported throughout the cell for food. The plant cell, right? So the plant cell has the chloroplast, and this kind of shows that whole process. Look, we've got in the chloroplast, we've got water going in, water is going to become split, the oxygen leaves, the carbon dioxide enters, and then there's interaction between the chloroplast and the mitochondrion. So remember, plants also have a mitochondrion. Okay, so the chloroplast is going to produce the organic molecules, which will then be used in cellular respiration to break down to obtain energy for the entire cell. Remember, the cell needs ATP. You can see the ATP coming out of the mitochondrion in order to do all the cellular work that cells do. So this could involve transport of things in and out of the cell. It could also involve replication of the cell, like movement of things across the cell, DNA replication, right? So all of these things kind of depend on both within the plant, depend on both the chloroplast and the mitochondrion. And these are the related chapters. So we've already discussed transport across the cell. Remember, pumps require energy from the hydrolysis of ATP, discussed in chapter 7. And then, of course, this unit discusses all the energy transformations in chapter 8, 9, and 10. And then this kind of shows, future things that we'll be discussing. So here I just talked about the chapter 7, 8, 9, and 10. However, energy is required for the flow of genetic information, which we'll be getting to. So we kind of introduced this, the production of these molecules and macromolecules in chapter 5, and then production of all the organelles and what the functions of all the organelles are inside the cell. And then also in chapter 7, the movement of things in and out of the cell. And then 16 and 17 deal with DNA replication and gene expression. So again, all of these things require the energy that is produced from the mitochondrion, right? And then, of course, in order to do that in a plant cell, the chloroplast is required to make those sugars to use for energy to do all this work.